Thank you, Ivan. And one of the very few good things with COVID is that I'm at my house in Israel, in the other side of the world, being able to join you in the Latin American meeting. Before I will start, I want to acknowledge Shlomi Aaron, a student, a PhD student in my lab, that while he was doing his master degree with Professor Yael Lubin and Merav Seifan, we were doing together this survey of arachnids in caves in Israel. And Igor himself also helped us to identify and sort the material. So when talking about caves, we usually mean subterranean habitats which we as human beings can enter. But for many organisms, including arachnids, many voids, small or large, can be used as habitat, and it doesn't matter if it's natural or artificial, as you can see here in the picture. What is uniting all these subterranean habitats are special characters, such as the limitation of light that is found in all caves around the world. Subterranean habitats, caves and other habitats, as I said, around the world share these unique conditions, such as lack of light, but also nutrient restriction. And you can see in this very nice figure from Stefano Mamola paper, a cave. While you enter deeper to the cave, the sunlight and the trophic resources are diminishing, but also the climate is more stable. And this is a very unique system. These abiotic and biotic conditions, sometimes with fragmentation and isolation in some caves, can, together with ecological filtering and evolutionary process, lead to specific local assemblages and act also as strong filters on the regional species pool. So, for example, if we see here a regional species pool of 10 species, only 5 of them can pass the abiotic filter of the specific habitat of the cave. And from these 5, only 3 pass the biotic filter to form, this specific assemblage. So selective pressure resulted from these special conditions, as I showed you, can lead to physiological, behavioral, and morphological adaptation, such as deep pigmentation, as we can see here, and even eye reduction to complete loss of eyes. We can define actually groups of organisms in caves according to this morphological adaptations, life cycles, and the phenotype. Troglobites are obligatory cave dwellers that complete their life cycle only in caves, and most of them have these adaptations, while troglophiles have pre-adaptation to life in caves and make complete life cycle in the caves, but can have also populations outside of the cave. I forgot to show you. Usually we find the troglobites in the deep zones of the cave, while the troglophiles are found sometimes also in the deep zones, but mainly in the twilight zone, and several other groups of organisms that can be found in caves, but usually in the cave entrance have no adaptations to caves, and they cannot complete usually their life cycle in the cave. So looking at this, and in Israel, even that in areas of the world, some of the subterranean habitats are very well documented. In the Levant, this is not the case. And therefore, we wanted to do a preliminary or first survey to see which species are found in the caves in the Levant. But maybe first it's important to say what is the Levant. The Levant is a biogeographic unit that is composed of northeastern African and northwestern Arabian plates and the eastern Mediterranean Levantine basin. And its geographical location in the junction of three continents, Africa, Asia, and Europe, and the diversity of habitats and climate zones, Mediterranean climate, as you can see in some of the pictures here, and the steppe and desert habitats, as you can see here down in the slide, laid the foundation for diverse regional species pool. Also, the historical geological events contributed to the formation of extensive and diverse subterranean habitats in the Levant, such as primary and secondary caves, different rock types, and also caves that were formed below and above the water table. So we aimed at, as I said, discovering the arachnofauna of the Levant and describing it, but only anecdotal sampling in caves in Israel and Palestine were done before, and due to the high diversity of cave habitats in our region, we first wanted to design a survey to include representative of different cave types. We hypothesized that cave size, bats found in caves, and if it's insectivorous or frugivorous bats, as well as the geographical region, if it's in the Mediterranean part of Israel or the desert part of Israel, this will shape the assemblage composition in the specific cave. Between 2013 and 2015, we did the first survey with around 55 caves around Israel and Palestine. Among these 55 caves, 33 caves were selected to do survey or monitoring by protocol when we tested for with visual search 10 minutes in the out of the cave in the entrance, but out of the cave in the entrance in the cave, the twilight zone and the dark zone of the cave. Of course, according to the size of the cave, small caves had only entrance. This is a time to acknowledge also Marco Ijaya and Stefano Mamola, helping us with discussing how to do this survey, and Bernard Huber that joined us to the first survey. We, of course, looked for all arachnids and other arthropods we found, and we tried to have even a set of caves with 3 to 4 caves, large caves, 3 to 4 medium caves, and 3 to 4 small caves in the northern part of Israel, central part of Israel and Palestine, and in the south of Israel. So this is just very few of the very amazing diversity of arachnids that we found in caves. To sum up, we found 21 or actually 22 spider families from 55 families known, spider families known for Israel. We found many new records and taxa. Some of them were already described. Some will be described soon, I hope. 3 new families of spiders to Israel, and new records of opiliocarids, a new family of opiliones, Palpigradi, and many pseudoscorpions. So a lot of work still to be done. If we look at the around 720 individuals we collected in this specific 33 caves, in this specific survey, you can see the rank abundance of the spider families, where the black column are families with troglobite and troglophile representatives. The gray columns have the troglophile representative, and in green, these are mainly occasional visitors, families with occasional visitors in the entrance of caves. Overall, we had 11 spider families with affinities, some of them very strong affinities to caves. If we look at the geography or geographical regions that are corresponding to the climatic regions of Israel in the north Mediterranean and in the south desert, although we had a similar number of caves and very similar effort of search, of visual search, almost 50% of the individuals we found were from caves in the north of Israel, while only 16% were from caves in the desert of Israel. You can see here in the graph again rank abundance. In red, you have the overall total number of spiders according to the family. In green, this is for caves in the north of Israel, blue in the center of Israel and Palestine, and yellow in the south of Israel. And you can see here very nicely that caves in the north of Israel were dominated by Arachnids, Sicarids and Philistatids, and also Phorcids, while in the south of Israel, the caves were dominated mainly by Theridids, probably one species, Teotoda triangulosa, a few Arachnids, and also several species of Phorcids. And in the center of Israel, we had species that were found both in the south of Israel in the caves and in the north. Some families were found only in the caves in the north of Israel, and also some species, of course. In this graph, you can see a presentation of the RDA ordination when we tested the bats, if there is, what is the effect of bats in the caves and the type of the guano, if it's a frugivore bat or insectivore bat, the geographical region and the cave size. And you can see very nicely in the lower part of the graph, all caves, you can see in the graph is a very good separation between the caves in the northern part of Israel and the caves in the south and eastern part of Israel, here in the upper part of the graph, and also a very nice separation between large caves and the rest of the caves. We can see also separation of very few caves that have frugivorous bats, and their assemblages are totally different from all other caves. What is also interesting is that in the caves here were mainly the caves, the medium and small caves of the south of Israel, the desert, but also several caves from the eastern part of the center of Israel. They were very similar in their spider assemblage.
Sorry, Efrat, that was 12 minutes.
Okay, I'm 2 slides and finishing. So Israel and Palestine have a rich subterranean arachnofauna, and we have ongoing taxonomical and faunistic efforts. Regional species pool is strongly filtered by abiotic habitat conditions as the climatic regions, but also guano may have a strong bottom-up effect on cave assemblage via food web, and we are very happy to test it now with a new postdoc in the lab, Jordan Kaff, and another PhD student, Zina Ghanem. And also just very shortly to tell you about all the different interesting research that we do as a result of this survey, mainly in collaboration with the lab of Professor Prashant Sharma. We're trying to understand what are the genetic mechanisms of eye reductions in arachnids and what are the origins of troglobite in Levantine caves. So thank you very much. And if there is time for questions, I will be happy to answer.